Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. We continue our lecture from the topic cell culture. Right? Cell culture is one important part in the biomedical engineering studies. So it is a technique what we use to remove organism and place in the fluid medium, but we need to look at the proper conditions so that the cells can live and even grow. So the growth can be categorized by cell division, mitosis, or by other processes. The term cell culture refers to the culturing of cells derived from multicellular eukaryotes, especially animal cells. However, there are also cultures of plants, fungi, insects, and microbes, including viruses and bacteria. So there are many types of them. Right, let's look at the application. The application of cell culture are quite vast. Cell culture is the what we call major tools in the studies. For example, cellular molecular biology, study of normal physiology, biochemistry, the effect of drugs, toxic compound, mutagenesis, carcinogenesis, drug screening, development, large-scale manufacturing of biological compounds. So what is the major advantage of using cell culture? Right, so if you use cell culture, that means we are testing our whatever the compounds, the drugs, and some sort of studies in the what we call cell lines or group of cells. So instead of testing on animal, testing on human prior to that, so we do test as a cell line based study. So it is consistent and reproducible. When you use studies, cell-based studies, it is consistent and also reproducible, and the result obtained from this what we call batch of clonal cells. So that means you can repeat the studies 5 times, 6 times, or as many as you want, and you will cause no harm to for example like animals or human, but this is in the cellular base of studies. Right, among there are so many cell culture cells that been used. One of which that we will focus in this topic is called Hela cell. Hela cell. So Hela cells is cervical cancer cells. It's a cancer cell. So it is quite what we call famously used in many cell culture studies. Right, this is how the Hela cells look like. If you look at this figure, this is how the Hela cells looks like, and this is a photo of taken by optical microscope. So the culture Hela cell procedures, apart from taking from organs and tissue, this is the second stage already. This is second stage where we have the cells is already in the flask. Cells already in the flask. I mean, this is the culture flask. Cell is inside the flask. So prior to remove them, you wash them with phosphate buffered saline (PBS). So you rinse them, and then you detach the cells. So when the cell is stick on the culture flask, we cannot simply just remove them, but there is a certain procedures. This is the procedure. First, you rinse them, and then you remove the fluid. Then now you add this trypsin inside the flask, maybe 5 to 10 ml of trypsin, maybe around 10-15 minutes. So before adding trypsin, the cells looks like this. Once you add the trypsin, the cell all will become like this. So they are rounded in shape. They're rounded in shape because they are protecting themselves from this what we call trypsin. This is the types of media that you use to remove the cell from the surface. After that, you monitor them in the microscope so whether it is fully detached or not, and then once it is fully detached, you add some media and then you incubate them, right? If you incubate them, that means you after that you will do a centrifuge, you spin the cells and remove the trypsin plus media, and then you resuspend them in the fresh media, and then you plate them again. Okay, this is what happened after you have removed the trypsin and growth media, and you have plated the cells again. Let's say when you first plate the cells inside the flask, it is very minimum number of cells, so it is this image is taken 6 hours after the cells been plated on the flask. Then you have 12 hours, 18 hours, 24 hours. So by 24 hours you can see how the cell is well attached to the surface, so it is clear. Then you have 30 hours, if you monitor them, another 36 hours, 42 hours, and then 48 hours. You can see there are so many of them are growing. But if you want to split them, and also you want to conduct the process of replating the cells, that means cells behavior is that they will find the free area. This is, for example, like the free area. So the cells will try to attach on this area. If they are already attached, they will try to move to the free areas. Once there is so many number of cells on the surface, so they will start to fight for new areas. But if there is no more areas, that means the cells already fully grown. That means 80 to 90% confluence level, such as like this in the image. So now we can remove the cell. We have to remove the cell. If you don't remove, you will start to see some of the cells already what we call clashing with other cells, and then they will be floating on the surface. This is called they are dead cells. So if they are dead, they will start to float, and you will see so many number of cells floating on the media. So this is what will happen if you don't resuspend them, transfer them to a new flask. So you have to repeat again the trypsinization process, and then replate them inside the new flask. Okay, moving on. Now let's look at the application of this cell culture, where we will use the cell culture. So one of the area that is being focused is called cell engineering. So basically this is a area that will lead you to tissue engineering. So before we go for tissue engineering, normally we will start at cell engineering. Cell engineering is basically we as an engineer, we try to guide the cells. I mean, you guide the cells so the cells will move in the direction that we want. So for example, if we have a wound area, so how are we going to guide the cells so that the cells move in the direction of the wound, so that the wound can be closed? So this is one field from the medical and also biomedical engineering, right? So these, before we go to the tissue level, as I said, so we have to study at the cellular level, subcellular level, so that this area has emerged to become a new area, which is called cell engineering. Cell culture technology, the ability to grow living cells in artificial environment of laboratory. This is what we looked at before. And then engineering includes the roles of engineering both basic cells biology, research, and in the making of product which use living cells, tissue engineering, and also bioprocess engineering. I have selected one studies. For example, in this figure you can see this is called the high voltage pulse generator system, ECM 830. So this is how the system look. Okay, the company name is BTX. So and then they have this chamber, and then this is the cuvette that we will include inside the chamber. The chamber will be connected to this system, and we will use this system to excite whatever is inside this cuvette. So inside this cuvette is, we will put some cells in there. For example, if we have Hela cells, so we will put Hela cells inside there, and this cuvette will go into this BTX chamber, and then from this chamber is connected to the generator. So what is this ECM? It produces square wave pulse generator designed for in vitro and also in vivo application. I'm sure by now you understand already what is the difference between in vitro and in vivo. Vitro means outside the body, vivo means inside the body. The versatility of ECM 830 application, so they use for gene, drug, protein delivery, mammalian cells, in vivo, ex vivo, tissue, zebra, fish, tissue, and embryos, nuclear plants. There's so many studies, right? Can be used also in combination with a wide array of BTX specialty electrodes. So there are more modification can be done to the system as well. But this is the range, the features of the ECM 830 generator. Voltage, so you can vary them 5 to 3000 volts. Final voltage discrimination, pulse duration 10 microsecond to 10 seconds, arc quenching, and digital display of actual voltage and pulse length delivered. Okay, these are some of the applications that been used in vivo application. Okay, so the system, what we do with the system is actually, for example, if we have this is the cells, right? So you detach the Hela cells, and then you include the cells in the cuvette, and then you put in the chamber, and then you excite the cells with the ECM 830 generator. So that means we are passing through that whatever the voltage ranging from 5 to a few thousand volts. So this voltage will pass through the cells. That means this is called electroporation. So basically what you do is you excite the cells with electric field. Either it is a high voltage or low voltage. So I would like you to also look at the studies called electroporation. So there are two modes available. So with this system, you either can heal the cell or you can kill the cells. That means you use the high voltage pulse or low voltage pulse, so that you will excite the cells. That means it will create pores. The passing of electric field on the cell will create pores on the cell membrane, and then the cell membrane will start to expand, and the increase in the pore size, what will happen is that the surrounding media, the growth media, proteins, will easily enter to the cells and reach the cells. So that's one application, which is healing the cell. But and then at the same time, if you use the electroporation, the high voltage pulse generator, to excite the cell or with the very high voltage, that means you are going to kill the cells. So this is also been used to do treatment of tissue ablation and studies like that. So you have to decide, if you want to kill the cells, then you have to use a setting parameters which is called irreversible process. Irreversible, that means you cannot reverse the process. But if you want to heal the cells, you just excite the cells with the low voltage, and you can reverse the process. That means when you excite the cells will be swelling, and then at the same time, after a certain period of time, the cell will reverse back to its original condition or shape. So that's what we're trying to do here, so that we excite the Hela cells, and then we monitor the time taken for the Hela cell to grow. Okay, that's one kind of excitation. Another one is we can use the magnetic chamber for high resolution real-time visualization. This is the chamber. So in this chamber you can see there is two electrodes there. So these two electrode is connected to these wires, and then this wire is going to the ECM830, the pulse generator again. Okay, so and then you will place cells inside this chamber, within this chamber, with the glass slide, so you can monitor the process of cells growing and splitting and attaching to the surface through this microscope. So let's see here. This is what we call the setting for high resolution imaging system. That means you will put this electrode inside the dish, and then whatever the cells sitting in between this chamber will be excited by the high voltage pulse generator, and then we use this microscope system to image the cells in real time or time-lapse recording. For example, 12 hours, 24 hours of 48 hours. Then you will be able to see the cell movement in real time. This is just a diagrammatic representation to show the setup of the system. Right. That's two types of studies that you can do to look at cell movement on the surface. Cell movement, cells growth on the surface, cell splitting. So one is, I've covered so far, one is to use the chamber, cuvette. Another one is to use magnetic chamber, and you excite these cells with the ECM system. That means, like I said, you have to find more information on electroporation based studies, which is which consists of two types of mode. One is reversible and another one is irreversible. So the third one is surface micro patterning technique. So basically what you do is this process is also called micro contact printing. Alright, so you need the glass cover slips so that to attach the protein solution through this micro contact printing. So what you do is, this is the process mentioned here. Number one, you need this PDMS stamp. So it is silicon based, and then you ink the stamp with the proteins, and then you use the same stamp. After inking the protein, let the protein stick to the stamp, and then you stamp it on the glass substrate, and you put a small force, and then when you remove the PDMS stamp, you will have the protein aligned on the glass slide. Okay. So coming back to this studies, this is one particular application of cell guidance, right? It's called cell guidance. That means through this protein deposited on the surface, we can actually guide the cells. The cells normally the cells are very much interested in the proteins on the surface. So these proteins will attract the cells to be guided on the surface. So if you deposit proteins, that means the proteins will act as what we call macam apa nama dia, dia akan menjadi macam track, macam jalan raya. Okay, so wherever there is a protein, so the cell will attach to these areas only, and then they will start to move in the same direction. So just imagine if you print in the same direction of this protein, then the cell will start to follow. This is basically the idea. Like I told you before, when these cells is been guided, so they will move faster in the same direction, instead of when you see the previous image. If I can show you, for example, this, now you can see the cell is going everywhere, this area, that, so in every direction. But when you use the tracks of protein micro contact printed on the surface, so it's gonna move in the same direction. This is where you can guide the cells. So this is a third example on the cell engineering, right? And then this is the cell electroporation studies that I explained before. So basically you use the system to for example, like transfecting the physical or we call the cell membranes, through which substance like nucleic acid can pass into the cells. Alright, so basically this is again the process of how the electroporation works. So this is a comparison to show the advantage and disadvantage of electroporation. So the main advantages is you can use this method as a stable transfection of all cell types, because electroporation is easy and rapid. It's able to transfect large number of cells in a short time once optimum electroporation conditions are determined. That means if you have multicellular, that means so many types of cells, so you can use the system as the same method to transfect a large number of cells. You can do it on the group of cells and also on the single cell. Major drawback of electroporation is that substantial cell death caused by high voltage pulse and only partially successful membrane repair. That means sometimes when you do excite the cells, if the cells are dead, so it is quite difficult to recover the dead cells. So only the partially one is successful in membrane repair. So this is one of the drawbacks of the electroporation. But the idea of electroporation is that when you excite the cells with the reversible mode, that means you are actually exciting the cells so that they swell, and also at the same time they uptake all the medias, the proteins, and the important fluids for them to grow. So we have monitored some of the studies using electroporation in cell growth and also cell guidance. So I want you to again look at cell guidance studies based on electroporation, and also wound healing studies based on electroporation. So these two you must find some papers online, and also try to make notes on these two types of application. So if I give you application, give one or two application, you are able to answer the question by giving one is the cell guidance type of studies, another one is the wound healing process for electroporation. Okay, and that's it. Thank you very much for listening, and hopefully we will continue again with the next chapter for the next lesson. Wassalamualaikum warahmatullahi wabarakatuh.